হ্যালো, শ্যাম শোনা যাচ্ছে?
হ্যাঁ স্যার, গুড মর্নিং।
বলছি মর্নিং। আমাদের স্পিকার কি জয়েন করে গেছে?
হ্যাঁ, উনি জয়েন করেছেন।
আচ্ছা বেশ, তাহলে আমরা কিন্তু শার্প 11টাতেই শুরু করে দেব।
এক্স্যাক্টলি।
আমরা একটু ওয়েট করি, হ্যাঁ ছাত্রছাত্রী আর একটু জয়েন করুন, ঠিক আছে। নমস্কার, শুভ দ্বীপ্রহর। জুলজিক্যাল অ্যাসোসিয়েশন বর্ধমান আয়োজিত অনলাইন লেকচার সিরিজ ফর আন্ডার গ্র্যাজুয়েট স্টাডিজ ইন জুলজি, এই শিরোনামে প্রাণীবিদ্যা সাম্মানিক পাঠ্যক্রমকে সামনে রেখে আজ আমরা দ্বিতীয় বক্তৃতার আয়োজন করতে চলেছি। আমি উপস্থিত সকলকে হার্দিক অভ্যর্থনা জানাই। আমাদের এই প্রতিষ্ঠান 1990 সালে খুবই প্রতিকূলতার মধ্যে তার পথ চলা শুরু করে আজকে স্বমহিমায় মাথা তুলে দাঁড়াতে পেরেছে। আমাদের নিজস্ব ভবনের ঠিকানা হলো শরৎপল্লী, পূর্ব বর্ধমান। আমাদের নিজস্ব ভবনে একটি বৃহৎ গ্রন্থাগার আছে। সেখানে নির্দিষ্ট বিষয়ের জন্য অসংখ্য বইয়ের সাথে সাথে এই বিষয়কে কেন্দ্র করে বিভিন্ন বৃত্তিমুখী বিষয় ও তার সাথে পরবর্তী পর্যায়ে সংক্রান্ত পথগুলির দিশা নির্ধারক নানান পুস্তক দ্বারা সমৃদ্ধ হয়ে আছে। সর্বভারতীয় স্তরের পরীক্ষা নেটের উন্নত মানের প্রশিক্ষণের ব্যবস্থাও আমাদের সংস্থার দ্বারা নিয়মিতভাবে আয়োজিত হয়ে থাকে। আমি উপস্থিত সবাইকে আমাদের এই সংস্থার সদস্যপদ গ্রহণের জন্য হার্দিক আমন্ত্রণ জানাচ্ছি। আমাদের আজকের বক্তা খড়গপুর আইআইটি সংস্থার স্কুল অব বায়োসায়েন্সের সহকারী অধ্যাপক প্রফেসর অভিজিৎ দাস। তার বক্তব্যের বিষয় হলো স্ট্রাকচার অফ নিউরন, রেস্টিং মেমব্রেন পোটেনশিয়াল, অরিজিন অফ অ্যাকশন পোটেনশিয়াল অ্যান্ড ইটস প্রপাগেশন অ্যাক্রস দ্য মাইলিনেটেড নার্ভ ফাইবার্স। এই বিষয়টি পাঠ্যক্রমের সেম 3 এর ষষ্ঠ পত্রের অন্তর্ভুক্ত। আমি বর্ধমান রাজ কলেজের প্রাণীবিদ্যা বিভাগের মাননীয় সহযোগী অধ্যাপক ডক্টর শ্যামদাস বন্দ্যোপাধ্যায় মহাশয়কে আজকের বক্তাকে উপস্থাপিত করার জন্য অনুরোধ করছি।
Thank you very much. Good morning to everyone. It's again a pleasure to have you all around for this interactive session. As you all know, being organized by the Zoological Association of Burdwan. Today, we have Dr. Abhijit Das, an expert in chromatin biology and epigenetics, currently associated as an assistant professor in the School of Bioscience, Indian Institute of Technology, Kharagpur. Also happens to be a former member of this esteemed association. Dr. Das was born and bred in this town, who pursued his bachelor degree in zoology from Vivekananda Mahavidyalaya, Burdwan, follow on masters in zoology from Banaras Hindu University, and then he completed his doctoral degree from the National Center for Biological Sciences, popularly known as NCBS TIFR Bangalore, one of the premier institute in India. He is an alumni of Garden Institute, University of Cambridge, United Kingdom. He was the recipient of several prestigious awards, including Ramalingaswamy Re-Entry Fellowship from the Department of Biotechnology, Government of India, and Fellowship of Indo-Swiss Bilateral Research Initiative, École Polytechnique Fédérale de Lausanne, Switzerland. Dr. Das has authored and co-authored numerous publications in peer-reviewed journals, which are of international repute. Let's give him a big hand and begin with the session. Please, Dr. Das.
Good morning, everyone. Thanks a lot, Sam, for introducing me in such a nice way, and thanks to a big thanks to Zoological Association of Burdwan for inviting me to give this lecture. So with that, I will start off immediately because we have very small time and we have to cover a lot of things. So yes, I hope everyone can hear me. Every student, whoever is present, can hear me. Sam, am I audible?
Yes, you are audible.
Okay, thank you. And my screen is visible, I hope.
Yes, it is.
Okay.
Please go ahead.
Okay, so to everyone, so this is the so the topic has been introduced already. What I am going to cover today. So this is solely targeted towards you, the students, the undergrad students, whoever is studying in semester 3 or whatever semester you are in. So I'm sure you can gain something from this lecture. I'll be more than happy to take you through this topic, and maybe some of you already know the topic, some of you are slightly familiar, some of you are totally familiar, but you are very much welcome to ask me questions, interrupt me in the middle, of course keeping in view the total time we have. Okay, so so let's start with the lecture. So I have titled my total lecture in this different title, that is Fundamentals of Brain Function. So what we are going to learn today is the enigma of the brain, but of course we cannot cover the whole topic. That is probably a very thick book that it will be. So instead of that, we learn the very basic of the brain function. So this is our brain. Of course, it's a simulated version, and what we do today or every day is totally dictated or guided by our brain. As you are listening to the lecture, watch a movie or listen to a song, memorize something, remember your old past memories, and analyze everyday events to take necessary actions. Every action of your day-to-day activity is guided by the brain. All your physiological, metabolic activities are there that your body is performing anyway, but your brain is dictating almost everything. So that is one of the most complicated science to understand the brain. It's said these days that if there is one science that is most complex, that is equivalent or more complex than rocket science, that is neuroscience. So how we sense our environment and how we take necessary actions, those are included under this topic called cognitive science or neuroscience. So of course, we'll study the very basic of it, and the fundamental of brain function, of course, is dictated by, again, neurons, a very tiny cells, just like any other cell of our body, and these are the structural and functional unit of our brain. And these cells, to study them, your syllabus has these given topics, that is structure, to understand the structure of neuron, to understand how they function, and that is divided into these subtopics called resting membrane potential, origin of action potential, and how this action potential propagates across different types of nerve fibers. So in order to start that, I will take you through very basic of life science. So what is the characteristic of a living system? Let's understand that first, and that will make our job easy. So I'm sure you appreciate that we all are very highly complex structures in the universe, and that are evolved over the course of evolution, and we have organized a system for extracting, transforming, and using energy from the environment. We can adapt, we can evolve. One of the very basic tenet of life is ability to develop and grow, and of course the capacity to recapitulate ourselves so that we keep a copy of ourselves in the form of progeny. And the last, not the least, and I believe one of the very important one is the mechanism of sensing and responding to our environment. So here we are talking about how to receive signal from outside and how to send signal outside, and in the process modulating the signal. So that's what our brain does. So our brain is just for sensing the environment and responding accordingly and taking action. How do they do so? So they do so by the structural and functional unit of our brain, that is called neuron. Of course, a neuron is just one unit. We have billions of neurons in our brain, but a neuron's function, if you understand that, we'll understand a lot of the brain function. So let's see what a neuron looks like. So this is just a typical cell that we have studied, that we know that what it is composed of, how it looks like. This is a typical animal cell. Most often we draw it as a sphere or a circle, and then inside it we put a lot of things, a lot of tiny dots, big circles, these subcellular organelles. But unlike a typical cell, a neuron looks quite weird. It has a particular very different shape compared to a typical cell. At a glance, it doesn't at all look like a cell. So it has a very quite large, almost like a sphere cell body. So that is, you can compare that with the normal animal cell. Beyond that, there are many branches that the cell forms. So these are nothing but hands and legs of the cell, if you want to call it like that. So these are tiny branches which are called dendrites. I'm sure these structures you have learned from your childhood, from class 7 onwards, I guess. So these are, we are not learning anything new from there. So these are called dendrites, which are the signal receiving units of these neurons. Then there is a large or long tail-like structure which is called axon. This axon is the part which projects from the cell and then goes and connects to something else. This something could be another neuron or it could be another muscle. So it can receive signals from dendrite, process it somehow, and then take it and send it to another neuron or to another muscle. By that process, so you can already appreciate that this is the function of our brain also. Receive the signal from somehow and then send it to our muscle so that we can take necessary action. So in this neuron, we have sometimes this neuron's axon part is coated with a special type of sheath or covering, which is called myelin seed. They are of huge importance. We'll learn about that later. Okay, so this is just one neuron, and that is, you are seeing it, that it is connecting to another neuron. So neurons, as you have seen before, that this is the structure. It has one signal receiving end, another signal sending end, but that is very typical neuronal cell. There are multiple variations of that neuronal cell, as you can also appreciate that how much variations are there of typical animal cells. Each cell of our body looks different than this one. So neurons are also not exception. They also have variations. So this is the one we have learned so far. That's the unipolar typical neuron. That is, unipolar means here based on how many branches are present in which side, that dictates what type we call them. So this is unipolar. That is, it has only one pole. The bipolar, it has two poles. The cell body sits in the middle and two long branches start from that cell body. Pseudo-unipolar. This cell body sends one branch that then can form multiple branches. So it's like one pole, but it's not really one pole. It has multiple poles. Then there are real multipolar cells where the cell body sends multiple projections, which are dendrites. So it has multiple endpoints or poles from which it can receive signals. And then there is another end which sends the signal to another cell. So in this way, you can divide neurons, but of course, there are multiple other variations of these subtypes. We're not going to that detail. Instead, we'll go to the functional aspects of neuron. That is most interesting, and that's how we can make an attempt to understand brain function. So what is so special about neurons that make them neurons, that make them most crucial cells of our body? That is, in one sentence, you can define that as neurons are the cells which can receive and transmit information. That's what they specialize for, and let's see what is that specialty. Again, let's go back to very basic before we go to neuron, because this is very important to understand. So this is a cell's membrane. I'm sure you have studied cell membrane structure. If you're not, I'm sure you will, in a few months, you will be learning about that. So this is a cell's EM structure. You can see very well that there is a covering outside a cell. That covering is called cell membrane, which is composed of nothing but lipids and proteins. These red human-like structures are the lipids. These are kind of making a coating on the cell and making it semi-permeable, so that not everyone can enter the cell, not every molecule can exit the cell. So these form a barrier. In the barrier, there are some other structures studded within that bilayer. So this is kind of a 3D structure of a piece of the membrane, in which you can see that membrane proteins are located. So these membrane proteins have huge significance. In our context, they have very specific function in neuronal function. So let's see what those are. So this, before we go there, go to understand these proteins function. So let's talk about differential permeability of the membrane. So as I said, the membrane is not permeable to everything and anything. Instead, it can block entry or exit of certain molecules and allow certain other molecules. These arrows are telling you how easily these hydrophobic molecules can pass, but these ions, on the other hand, because they are charged particles or the large particles like glucose or sucrose, they cannot pass so easily through the membrane. So these are called, these are effectively blocked by the membrane. But our cell has to take up ions, has to send ions outside. So how do they do that? They do that by means of special type of proteins that are present in the membrane. So these proteins are called channels. So these channels kind of form a pore across the membrane through which this charged molecule can pass, which are otherwise blocked by the hydrophobic membrane. Right, so these channels can be of various types. And of course, one major property of these channels, the channels that we are talking about in the membrane, are very selective. And they can connect the cytosol to the cell exterior. That is, they are allowing things to pass which are otherwise non-permeable. Many channels are there in the membranes which are ion channels. Remember, we said that ions are not, they find it very difficult to pass through the membrane. So there are many ion channels which allow ions to pass. A very beautiful property of these channels are they are gated. They can be opened or closed, just like your house gates, right? So, and this channel activity can be very easily regulated by the modification of the channel proteins. These are proteins, nothing but amino acid sequences. So you can imagine that they can be modified or modulated. And advantage of these channels are that they are very fast. Disadvantage is that they cannot be coupled to energy source. That is, they are only allowing ions when there is a gradient. So what is gradient? We will come there, but let's talk a little bit about the ion channels. So there are different types of ion channels. One is leakage channel. So whenever there is a ion concentration gradient, that is if there is more ion inside compared to outside, these channels will just leak the ion. That is, this is allowing the ions to pass so that there is a balance of ion numbers across the cell membrane. Then there are gated channels. I was talking about gates. So here there are gates. Channels are not always open, always closed. They can be, according to need, opened or closed. So now this gating can be of various types. Some are gated by voltage, depending on what is the voltage difference between inside and outside. And what I mean by voltage difference, I'll come to that. So there are other types like ligand-gated ion channels. For example, a protein can bind to the channel and effectively open or close the channel. By doing so, it allows our ions to pass or blocks the ions to pass. So there are different types. I'm not discussing too much about them. Let's go to other channels like mechanically gated ion channels, which can open or close depending on the different mechanical stimulus. So for today's topic, we will be mostly concentrate on these two types of channels, which one is called leakage channel, another is voltage gated. So this is showing you, the gray bar is showing you the kind of the membrane, and across the membrane there is a concentration gradient of ions. So this, and in the middle, this channel is sitting, which would allow the ions to pass, but only when this channel is open. So this is voltage gated channel. Once a certain voltage difference exists between inside and outside, then it opens, and then the ions are allowed to pass. Second is the ligand gated ion channel. This is the blue thing is the channel which, when binds to a particular ligand, it could be a protein, it could be a small molecule, but on binding to that a specific ligand, it opens up so that it allows ions to pass. Similarly, there are other channels. I'm not going to discuss about them. You can read up with the reference I'll be giving at the end. So this is one very important part before we understand neurons. So this is telling you what is a gradient. So by gradient, we have observed what gradient means. If there is a difference in concentration in inside versus outside of a cell. So this is again, the gray bar is a membrane. In between, in the membrane, there are multiple proteins sitting there. So this is, and these arrows are showing whether the ions are passing or not. Okay, so these are, and in between these ion channels are sitting. So this is showing you multiple molecules as a cartoon. So these round or square dots that you are seeing in different colors are signifying that there are more ions on this side of the membrane compared to this other side of the membrane. So there is a steep concentration gradient of these boxes or the round structures. So there will be always a gradient which is asking these molecules to pass through the membrane. That is the basic or fundamental way a diffusion happens, right? But through the membrane, diffusion always cannot happen because these are ions. If they are charged particles, they cannot move across. So there is a concentration gradient. So what? They cannot pass. But if there is a channel which is allowing them to pass, then they will go. But go in what direction? That is dictated by this concentration gradient, right? So if this yellow molecules, which are there are 3 outside and 1 inside, then if this channel is allowing them to pass, the yellow is always going to pass from outside to inside. Till when? Till the concentration gradient is dissipated. If there is no more gradient, this will stop. This passage will stop. So now that is concentration gradient. But what if we have to also consider the electrical gradient? When they are passing, they are not only passing molecules, they are also carrying charge. So that is also causing the charge distribution difference between inside and outside of the membrane, right? So we have different types of ions inside our cell as well as in the extracellular fluid. For example, sodium, potassium, or chloride ions, and their asymmetric distribution around the membrane causes a ionic difference. The charge difference between them. And the charge difference, if you consider in isolated manner, that also forms a gradient. And that gradient is not concentration gradient only, that's an electrical gradient. That is, there might be an effective net positive charge outside compared to inside. So it will be outside will be more positive than inside. So that's what is being shown here. Outside, if there are more positively charged ion, across 2 sides of the membrane, there will be a charge distribution difference, and that will cause an electrical gradient to exist. Now, on top of that, you can also consider chemical or concentration gradient. Together, they are called electrochemical gradient. Electrochemical, charge distribution and chemical distribution difference. Together, they are called electrochemical gradient. So you have to keep that in mind when we are considering membrane function in terms of neurons potential difference. So what is a membrane potential? So this potential is arising when there is a difference in the electrical charge on the 2 sides of a membrane. Remember, that is just generated by ionic concentration difference between 2 sides. And because they are carrying electrical charge, there is electrical charge difference between inside and outside of a membrane. They can result from multiple things. So here we have to consider multiple things when we are considering how this membrane potential is membrane potential is generated. So they are resulting from passive diffusion of the ions by the channels that we have discussed, as well as there are multiple types of pumps. When you study about membrane, you will learn that there are multiple types of pumps which are pumping ions from inside to outside or outside to inside. So these are ATP utilizing pumps which can pump different types of charged particle from inside to outside. So that is leading to generation of this charge difference. So initially, suppose there is no charge difference. So here we are now discussing how this ionic imbalance happens. So suppose there is this no charge distribution difference between 2 sides of the membrane. So suddenly, suppose there is a small flow of the ions from one side of the membrane to outside. Why would it happen? Because inside our cell there are many organic molecules, for example proteins, amino acids and others, which are negatively charged. They effectively drag the positively charged ions from outside to inside. So what happens is there is a net flow of positively charged potassium ions mainly from outside to inside of the membrane. That leads to, so this is causing the potassium ions to move this side. And when they are there, they are also willing to attract the negatively charged ion from other side. So in this way, positively charged ions will be lined on one side of the membrane, and they will form an electrostatic attraction for the oppositely charged ions on the other side of the membrane. In this way, there can be an imbalance between charged particles. For a normal typical cell, these are the different types of ions which are distributed asymmetrically across the membrane. So you'll find sodium is present more on the outside. So you will see that cytoplasmic concentration is much lower compared to the extracellular. So there is more sodium ions outside compared to inside. So what will happen? There is always a steep concentration gradient between its outside and inside. Lots of sodium is waiting outside to enter the cell, but they cannot enter the cell. Why? Because they are impermeable, and there are not many free sodium ion allowing channels which will allow sodium to pass in. So sodium is always kept more outside, less inside. On the other hand, so potassium is just the opposite. Inside our cell, there is more potassium compared to outside. And potassium always forms a gradient from inside to outside. So sodium and potassium always forms an opposite gradient, potassium more inside, sodium more outside. We have to remember this for understanding ourselves. And these are other different types of ions whose distribution is also forming gradients. So we can understand that there is a difference in potential or difference in charge distribution between inside and outside of a cell. And here we have to consider how that can change. And fundamental of neuronal function is nothing but dependent on this membrane potential. That's how it works. Let's see what are happening here. So this is just what we have already discussed. Like cells have negatively charged organic molecules which are asking potassium to come in. So there are lots of potassium coming in. And how they are coming in, we have discussed that there are leak channels in the membrane, and only type of leak channel that is present in plenty are the potassium leak channels. That's why when the inorganic molecules are attracting potassium can freely move in. And when they have moved in, they stay inside after reaching a particular concentration. And that concentration is called potassium equilibrium concentration. So we could go and discuss a little more, but at this point, I think that will be time consuming. So if you are interested, read a little bit about potassium equilibrium concentration and how they are generated. Okay, so that is one of the homeworks that you can do. Then at this equilibrium condition of potassium ion, there is no net flow of ions happening across the plasma membrane. So some potassium ion has gone inside, it has reached an equilibrium, and then no net flow of ions is happening. Of course, some ions are going in, going out, but net flow is important. That is, no extra charge distribution change is happening at this point. And that point, when it is reached, it's called resting membrane potential. So at resting membrane potential, a particular concentration difference between different ions have reached, and that is this, that is existing all the time unless changed. And that situation is called resting membrane potential, when there is a particular concentration of potassium present inside the cell, and that is causing a particular membrane potential to exist. And that membrane potential is called resting membrane potential. Usually all our cells have a particular resting membrane potential because all our cells have this plasma membrane across which ions can flow. But neurons are slightly more special because they have a particular ionic gradient, a particular resting membrane potential, which allows them to make a change in that, and that excites the cell. Okay, so before we go there, so this is the resting membrane potential of normal animal cell, which is variable between -20 millivolt to -120 millivolt, depending on which organism and which cell type. But in our neurons, specifically, the resting membrane potential is around -70 millivolt. But when we say minus and a particular number of millivolt, that means inside of our cell is always more negative compared to the outside. That's why it is denoted as minus 70 millivolt. How much difference? Minus 70 is the 70 millivolt difference between inside and outside, inside being more negative. Right, so now we'll go a little further from resting membrane potential and try to understand what is action potential. So I hope you have understood what resting membrane potential means. If I'm going too fast for the sake of time, please stop me now and ask me a question. So let me know what is your question about resting membrane potential first. Okay, your silence will be taken as you have understood everything, right? So your main, you may wait until the end and ask me questions at the end, but I'll be happy to answer. But this is the time when you can ask. Okay, so the resting membrane potential, let me remind you again, is just a state of the cell where there is no net ion flow is happening from inside or outside to the other side, and we have reached a particular concentration electrochemical gradient where the inside has more negative charge compared to the outside, and we have reached at around minus 70 millivolt across membranes. So now, how do we measure that? We can measure that by just poking an electrode through the cell. Keep one electrode outside, keep one electrode inside of a cell, and by that process you can measure what is the potential difference between them. Right, so this is nothing different than the type of experiments you do to measure potential difference. So here, let's go to the next step, that is generation of action potential. So this is a membrane. So this same cartoon will be repeated multiple times. So this is showing you a membrane. Inside, the yellow part is the cytoplasm. Outside, the green, sky blue is the exterior. In the membrane, you can see multiple types of channels sitting there, and these channels, one is showing as potassium channel, another is written as sodium channel. So this potassium channel is closed, and the sodium channel is also closed, right? So this is the resting stage. Here we are not seeing any leak channel. So these are another type of channel that we discussed. These are called voltage gated channels, right? So these are voltage gated channels, and they will be effectively opened or closed depending on what? Depending on voltage, because these are voltage gated channels. Unless a certain amount of voltage is, certain level of voltage is reached, they will not be opened. They are mostly closed unless a particular voltage is reached, right? And this sodium channel has a very peculiarity that this has two gates. One is activation gate, another is inactivation gate. So we'll discuss about that as the time comes. Okay, so at this point, this is resting state. And what is the potential difference between them? Inside is more negative, and that is 70 millivolt more negative than outside, right? So that's the state we are in. We are in resting state. So when we receive a stimulus from outside, this neuron gets activated or excited. How does it get excited? Just like you get excited upon looking at something great or something beautiful, similarly, a neuron can also get excited. And when it gets excited, it changes the ionic balance. That is, from minus 70 millivolt potential difference, suddenly it changes to something else. So let's see what happens. So this is, when that happens, it is called depolarization of the plasma membrane. The plasma membrane is having a polarity, right? Inside is negative, outside is positive, and there is a value minus 70 millivolt. And that is called polarization of the membrane, polarized membrane. So now, if that changes to a positive value inside or less negative value inside, that is depolarization of the plasma membrane. So that is the shift in the membrane potential to a less negative value inside. We had minus 70 millivolt inside. So let's plot that. In a resting phase, the resting state, we can just denote minus 70 as the RMP, that is resting membrane potential. So this has been denoted here as minus 70 millivolt. This is a plot of millivolt, that is potential difference, change over time. This will change when the neuron gets excited, when the neuron is stimulated. How does it get stimulated? We'll talk about that. But before that, let's see what happens when a neuron gets stimulated. So at time 0, it is minus 70 millivolt. Unless you give a particular stimulus to the neuron, it will keep maintaining minus 70 millivolt forever. But if you stimulate the neuron, things change. What will be the change? The change is called either depolarization, or the whole event that will be followed here are called action potential. So we'll talk about that later. Let's see what is happening. First, you stimulate the neuron, and that stimulation immediately changes the balance of it. What changes the balance of it? It goes up. In the y-axis you have minus 70 kept here, 0 would be somewhere here. So if it goes down, you will reach 0. That is you will go minuswards, 0 would be somewhere here. So if it goes up, we can reach 0, and then we can go plus 30. So what would happen? What would need to happen when we need to go up? That is when we have to increase the membrane potential from minus 70 towards 0. That is, membrane potential has to go up. What do we have to do? Can anyone answer me in a very short duration of time? What kind of change has to happen here?
So can you repeat the question, please?
So the question is, suppose I'm telling that here we have to change the membrane potential. The membrane potential is based on ionic concentration difference, right? So the ionic concentration difference exists between inside and outside, and that is dictating resting membrane potential. And here, let me draw it in a bigger way, but it's a tera way. So let's stick to that. This is minus 70 millivolt, but I'm saying resting membrane potential is keeping it like that. And I'm asking you how the membrane potential can go up. What has to change so that membrane potential would go up, would change to a more positive value? So
many gated potential has to be occur to uplift the voltage.
Yes, potential has to change, but what has to enter or exit the cell? I'm asking that. So there is ionic disbalance, ionic gradient. So ions can pass through, okay, through the channels. So these channels can open or close, but what type of thing can change? What type of ion can move in or out so that this minus can become plus? This inside cell we had minus, it has to become plus. So what type of ion has to move in?
Sodium.
Influx of sodium.
Yes,
Potassium,
exactly.
sir.
The K plus has to move in, and K plus has to move out, and sodium has to move in.
Yes, some of you have given right answers and some of you have given slightly wrong answer, but your idea is right. So the thing is, only one type of ion will move in. One type of ion can move in, that is plus ion. And in this case, it is sodium ion, because there is more sodium outside. Remember, we said there is more sodium ions. There are more sodium ions waiting outside just to be taken in. They are not taken in because this activation gate of this membrane of this sodium channel is closed, right? So this activation gate is closed, so sodium cannot pass in. So now when this action potential or the depolarization happens, it happens due to sodium channel opening and sodium moving in. If sodium moves in, our problem solved, that we are depolarizing the membrane. That is, from minus 70, it is going upwards, right? So action potential, the first step of generation of action potential is nothing but changing the gradient towards more positive value by allowing sodium to move in, right? So that's what happens here. Just give me a second. Yes, so when a neuron gets excited, it opens sodium channel. And let me tell you, these are voltage gated sodium channels. So you can immediately ask, if they are voltage gated sodium channels, they will be their opening or closing will be dictated by particular voltage. If they are at minus 70 already, at minus 70 they are closed. If they are at minus 70, if a voltage change has not happened, then how will they open it? So that is solved by how the neuron is first stimulated. So there are many ways to stimulate a neuron. So I'm sure at your kindergarten you have learned about 5 senses. We have 5 senses and 5 types of receptor neurons, sensory neurons are present in our nose, tongue, skin, ear, and eyes. They are stimulated by what type of stimulants? Our eye has photoreceptor, they get stimulated by light. Our nose has olfactory receptors, they get stimulated by some olfactory chemicals. Some good cooking is happening in your kitchen, that is coming in your nose, and those neurons, you can feel that there is a good cooking is happening because your nose neurons are getting stimulated by that chemical sitting on your neuron. So that chemical slightly opens a special type of channel, which are again sodium channels. So these sodium channels immediately open to take it slightly higher, and at this slightly higher it is about minus 55 millivolt. So when this minus 55 millivolt is reached by this chemical or light, whatever is sitting on your receptor neurons, they are slightly stimulated from minus 70 to minus 55. Some sodium ion has come in. When this goes to minus 55, and this minus 55 is the voltage, minus 55 millivolt is the voltage to remove this activation gate. This activation gate that I was showing here, that opens up at minus 55. So to activate this channel, you need to reach that voltage, and that is called threshold potential. So minus 55 is the threshold for opening the sodium channels, the voltage gated sodium channel. This is a voltage gated. So when they see minus 55 has been reached, they open. And there are hundreds or thousands of these sodium channels, all open and all allow huge amount of sodium ions to pass in. As they are passing in, this immediately goes up because more and more sodium ions are going in. This more and more plus ions are moving in, so the action potential goes up. And then, of course, sodium channel has another property that is called inactivation gate. So it gets inactivated at plus 30. So plus 30 is the voltage. When that is reached by movement of huge amount of sodium ions, immediately this and right here. Okay, so that is what is shown here in the depolarization state. In the depolarization state, sodium, this gate has been opened, sodium is moving in, and there is more sodium going inside. And see, these ions are showing to be, the charge distribution is showing to be inside more positive than outside. That is around plus 30 has been reached. When plus 30 or plus 50 in some cases, between plus 30 and plus 50, when it is reached, the sodium channels that had been opened before, now they are getting closed. There is no more sodium to move in, right? So that is called the peak of the action potential. So this whole thing that we are studying now, before this was resting membrane potential. Now we are studying what is happening to the neuron after it gets stimulated is called action potential. And this first part of the action potential is called depolarization. Okay, so this depolarization has happened because the polarity has changed inside the from inside to outside of the cell. So due to the polarity change, we have now reached plus side. So this action potential has peaked. After reaching the peak, sodium channel is closed. But that is not the only event that is happening at the peak of the action potential. Another thing is happening, that is called another ion channel is sitting here, that is called potassium channel. So immediately at plus 30 is the activation for signal for the potassium ion channel, voltage gated potassium ion channels. So these voltage gated potassium ion channels will open at plus 30, not just closure of sodium channels. So potassium channels are now opening. So what is the direction of potassium ion movement? There is more potassium waiting inside compared to outside. Remember, I said there are more potassium normally present inside the cell compared to outside. So lots of potassium will move out. As they are moving out, they are carrying positive charge. So inside will start becoming negative. So action potential is going downwards, right? So it is going downwards. So it's going down, 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 and it has come back to normalcy, to some extent normalcy, because this reaches beyond minus 70. So that is happening because lots of potassium ions are moving out, right? So here, sodium has come in, potassium has gone out, and when it stops, that stops because this voltage gated potassium channel can stop at minus 80 millivolt. Okay, so at minus 80 millivolt, this potassium channel will close, and no more distribution of sodium, no more rearrangement of potassium channel, sodium channel, so the ions will happen. So that's called the end of the action potential, but that is not really the end. That has taken the action potential to minus 80 millivolt instead of minus 70. So that minus 80 millivolt, because it has gone even beyond the resting membrane potential, that is called hyperpolarization state, and that is around, whose voltage is around minus 80 millivolt. Okay, so now two things happened. Sodium channels at the peak closed or inactivated, because this is the inactivation channel. So sodium ions are not moving out. Potassium channels, which opened at plus 30, are now closed at minus 80. So have we really reached the resting membrane potential? There are two things went wrong. One is, you can already imagine, minus 80 millivolt is not exactly same as minus 70. And another thing is different compared to the RMP, resting membrane potential. What is that? Here, we started with resting membrane potential, where inside had more potassium ions, outside had more sodium ions. But during this action potential, that reversed. Inside now has more sodium ions, outside has more potassium ions, because of this whole scenario, whole action potential. Though we have reached our resting membrane, nearly resting membrane potential, but the ionic disbalance, how will you dissipate that? That is a bit difficult, right? Now, these channels will not be able to dissipate that, because they are voltage gated. Before they reach a different action different level, they are not allowing sodium or potassium to pass through them. So something has to change. Who will do that? So now there is a different type of machinery present here in the membrane, which we are not showing. Those are called sodium potassium pump. So those sodium potassium pumps are going to make the change. So these are the sodium potassium pumps here, which pumps sodium out. Sodium ions will be moved out, and the same pump moves potassium in. So that pump makes everything all right. Sodium moves out, potassium moves in, and in the process, it actually pumps 3 sodium in one round. It moves 3 sodium outside and 2 potassium inside. So anyway, but by doing so, it also changes the resting membrane, the ionic disbalance. Sodium moves out, potassium moves in. The potential comes back to minus 70. Okay, so it remains at minus 70 millivolt. So that's how the ionic disbalance is dissipated, as well as resting membrane potential is reached. So that's how a neuron can get stimulated and then comes back to normal ground state. So several terminologies we have learned here: resting membrane potential, and then there are, so that is minus 70 millivolt. Slight change in membrane potential by some stimulants take it to minus 55. That is the threshold potential. That is number 2 here. From threshold potential, one type of voltage gated channel opens, that is voltage gated sodium channel. That makes the shooting of the action potential or depolarization to happen. Next, the peak of the action potential at 3, where two things happen: voltage gated sodium channels close, voltage gated potassium channels open. That leads to potassium 2, that allows potassium to move out, so that membrane goes through repolarization. That is, polarity is reverting back to normalcy. But it does not stop till minus 80 millivolt, which is called hyperpolarization. At hyperpolarized state, the neuron cannot fire again, and this moves back to normal ground state, minus 70 millivolt, by the action of voltage sodium potassium pump. This sodium potassium pump allows potassium to move in and sodium to move out. So that's how from hyperpolarization you reach a resting membrane potential. So here many things have to be considered. I'm sure it will take time for you to settle this understanding in your brain, but before that I have a couple of other things to discuss. One is, you are not asking one thing, that is, when this potential is coming back to normalcy, minus 55 is being crossed, but voltage gated sodium channels which opened at minus 55, they should have opened here, right? So there would have been another action potential happening here, but that is not happening, because voltage gated sodium channels have a special mechanism of inactivating for some time, and that is shown here. So here we are seeing that this is the voltage gated sodium channel, and this channel can sense the voltage when it is, see, this is the normal closing mechanism. It is closed at resting membrane potential. It opens when that goes to minus 55, so that this is opened. But when it reaches plus 30, this is not closing. The closing mechanism is not starting here. Instead, there is an inactivation mechanism started here at plus 30. That is at the peak of the action potential. This inactivation effectively closes the channel, as well as it keeps it inactivated for a long duration of time, so that it does not open immediately. And it is not opening immediately. That property is helping it to remain closed for a long time before it goes to a normal closed scenario. And that's how one action potential can complete normally, so that it does not produce another action potential here. That would have made a huge problem. Once you stimulate a neuron, it remains in hyperstimulated state all the time. So that it does not happen, this voltage-gated sodium channel has been evolved, and we are saved from continuously giving stimulation to all neurons once that is activated. Right. So that's how voltage-gated sodium channels prevent a neuron from firing continuously once it has started firing. Another thing that it does is, what we are studying here is just one neuron's action potential in one particular space. Right. So suppose we are drawing a neuron here. Suppose this is our neuron. We are not drawing too many dendrites or axons here. So suppose this is the cell body. So suppose this is just one dendrite whose this part is being stimulated. So this is the neuron which is getting stimulated. This is the part of the neuron which is getting stimulated. And when we zoom that part in, that is nothing but a membrane. And on the membrane, all these channels are there. So all this thing is happening inside and outside ions movement is happening on this tiny bit of the membrane. So what? That this tiny bit got depolarized and repolarized. The essence of the whole thing of a neuron is that it can spread, it can send the action potential throughout its membrane in this direction as well as in this direction throughout the membrane. Once it got stimulated at a particular point, upon receiving a particular signal, that signal is nothing but generating an action potential in that part of the membrane. Okay, so this part of the membrane has generated action potential. If we draw like this, suppose this is the cell membrane, which is a barrier between inside and outside, and action potential has been generated in this part of the membrane. Now it has to propagate. Now it has to transmit to other part of the membrane. How does that happen? So once there is a depolarization in this part, that depolarization can be sensed by the neighboring part. And the neighboring part means in the neighboring region, there are more voltage-gated sodium channels sitting there. So these voltage-gated sodium channels will sense that nearby there is a change in the voltage. So once this has started operating, the neighboring voltage-gated sodium channels also are facing voltage change, and that's why they will also open. So it will start propagating. Right. So once action potential has been generated here, depolarization has happened here, its neighboring sodium channels, these are also facing that engine polarity. So they will also open. And then they will see the polarity change. They will also open. So it's just because of the just because of being inverse, they will open and they will generate action potential as well. Right. So that's how action potential happens from one part of the membrane to the neighboring part. Now then there are other things that to discuss. So we have so far discussed that membrane, as if the membranes are bare, naked, they have nothing to wear. But instead our axons are coated with this myelin seed. They work like just an insulator through which ions cannot pass. So this part of the membranes are just like noded. So they have these coats, and in between coats there are some nodes. So when an action potential passes through these types of special type of fibers, which are present mostly in the mammalian systems, so once the action potential is generated, suppose in this part of the membrane, so that transmits, these are not myelinated, these are just normal membrane, action potential is transmitting like that. But here there is suddenly a roadblock. Action potential generated here will not be able to generate an action potential in this node, in this the myelinated part of the membrane, because these are insulated. Ions will not pass through the membranes. So what will happen is, once it has generated action potential here, it will immediately generate an action potential here. And that whose neighboring area where action potential can be generated is here. There an action potential will be generated. So from here onwards, action potential will be hopping from here to here, and that makes it much easier for action potential to pass. This is much faster conduction compared to the other parts of the membrane, where each part of the membrane has to generate action potential. Okay, so this type of conduction of the action potential is called saltatory conduction, or kind of a jumping action. This is a much faster way to transmit the signal through the neuron. Okay, so then again, it has reached the end of the action. Now it will transmit the signal to the next cell, and how that happens is a different story. And that, so far we have discussed about the chemical way of generating action potential, ionic way of generating action potential. Just the ionic balance changes and the channels involved were voltage gated. Next, what happens is it has to pass from one neuron to another neuron, but they are not connected. These membranes are not connected. So if there is an action potential generated here, it cannot generate an action potential to the next cell in the same way. So, but it has a different mechanism, and that mechanism involves some chemicals to be released from the first cell to the next cell. So these both can be neurons. The first one can be definitely a neuron. Second one can be a neuron or a muscle. And to transmit the signal from the first cell to the next cell, this mechanism is involved. Here, in this at the end of the neuron, which is called and the nearby vicinity, there is a second neuron or a muscle. So this type of structure is called synapse. I'm sure you are familiar with the name, and this gap between them is called a synaptic cleft. So in the synaptic cleft, there is no direct contact between them. So ionic disbalance or the action potential cannot be directly transferred. Instead, what happens is this action potential coming at the end of the axon induces these vesicles to open up to the exterior. So these are special type of vesicles of the cell. These vesicles carry a special type of chemicals called neurotransmitter. So these neurotransmitters, once the vesicles are released, these vesicles, once they fuse with the cell membrane, they release these neurotransmitters. So once these red dots or the neurotransmitters are released, their receivers or the receptors are present in the next cell, the signal receiving neuron or muscle. And these are nothing but, again, ion channels. These blue ones who are receiving the signal are called ion channels. So these ion channels are gated again. Normally they are closed. See, their gates are closed. It's not allowing ion to pass in. But once they are bound by these transmitters or the neurotransmitters, their gates open. So these are ligand-gated or neurotransmitter-gated ion channels. Right. So now plus ions can move in. It could be sodium. It could be calcium in many cases. So these different types of mechanisms are involved. We are not going to too much detail because this topic is not there in your syllabus, the part I am covering at least, and for the sake of time. Okay, so this is a huge mechanism involved here. That mechanism we are not studying, that is described here. And this is involving chemical substances being released to the next cell at the synaptic cleft. The next cell receives the chemical and opens plus ion channel. So this plus ion will move in and depolarize the next cell. And that's it. The next cell is also depolarized in a local manner, and that will spread throughout the cell. Okay, that's it. That's all I had to talk about. And these are different books or resources that you can follow. And I'll be happy to take questions from you. Hello, am I audible? I'm sure I have made a little, have taken a little longer, but I hope I have made the points clear. Any questions from anybody?
Students are requested to. Yes, please go ahead.
Yes.
Yes, sir. That I'm saying that how the resting potential reaches that threshold potential, that minus 55 millivolt, which
Yes.
ions exchanges occurs?
Yes. So the ion that is exchanged is sodium ion. In most cases, it is sodium ion. Okay. So there are two ways. One way I was telling that our sensory neurons, when receives those different substances like light or chemical substances for olfaction, so those are one mechanism to open some ion channels itself. So they are opening ion channels and allowing, in many cases, sodium to move in. But another way to initiate the whole process to reach the threshold potential is this. Can you see this slide? So here, another, if it is not a sensory neuron that is being stimulated, suppose the second neuron is not a sensory neuron. So, of course, is not a sensory neuron because it is receiving stimulus from another neuron. So it's a secondary neuron. So this secondary neuron usually gets stimulated by this method. It is receiving the chemical from another neuron. Upon that, it is allowing these channels to open. So what channels open here? I'm not showing you much, but this is what type of channels open. So usually it could be sodium channel to move in, and in many cases it is also possible that it can open chloride channels to open up. When chloride channels open, then what will happen? Can you tell me? So suppose this is showing that in the synaptic cleft, the second neuron has opened, where upon binding to the neurotransmitter, a different type of ion channel has opened. Here, chloride channel has opened. So if chloride channel opens and chloride passes in the cell, what will happen to the cell? Whoever asked the question, can you guess? I forgot who asked the question. Sorry. So can you guess what would happen when the chloride channel opens and chloride moves in? Just any guess. I'll not shout at you, definitely. I can promise. No guess here? What happens when sodium moves in? The cell gets depolarized, right? Aditya. Aditya, can you tell me, or anyone else can try what would happen if the second cell opens up chloride channel instead of sodium channel? Sodium channel opening will lead to sodium to move in, so that the second cell will get depolarized. When chloride moves in chloride channel, that is carrying negative charge, so the potential will go in what direction? Okay, let me draw it. Then you can guide me and tell me what is going to happen here. Yes, someone wants to try and tell me what is happening.
Sir, there is more negative charge inside the second neuron.
Yes. So let me, okay, don't mute yourself. So since you have opened up, so this is minus 70. So this is where we were. This second neuron was at minus 70. When more negative charges go in, which direction would we move? Upwards or
Minus
downwards?
80.
Yes, towards minus. So instead of making an action potential, it is going to downwards.
Yes, sir.
And when the potential is downwards, what is it called? Upwards, depolarization. Downwards,
Depolarization.
No, from upwards to downwards, that is depolarization. Hyperpolarization, exactly. So it is getting hyperpolarized. Hyperpolarization is happening here. Okay, if chloride is moving in, hyperpolarization will happen. So what is the necessity of having a neuron getting hyperpolarized? If a neuron gets hyperpolarized, now this neuron will be more difficult to depolarize. If it remains in hyperpolarized state, this will be more difficult to open up, more difficult to stimulate. Right? So that is also necessity in our body sometimes. Not just stimulating neuron, but to suppress neurons activity also. That happens when chloride channel is open. So suppose too much of stimulation is happening in some part of our brain. So there is a mechanism to stop that. Suppose you have seen something unpleasant and you have become too excited, and that too much excitement is not good for you. Suppose at that point that brain knows that. So it just inactivates your neurons so that you come back to ground state. You come back to resting state. So that mechanism is also there. Yes.
Sir.
Yes.
Sir, does the anesthesia depends upon hyperpolarization?
Kind of, yeah. It is a very complex mechanism, but to some extent you can say that it is kind of a hyperpolarization. Yes.
Okay, sir.
Very good point that you have made. Good.
So any question from anybody? So we have to conclude. Any question? We are running short of time. Any question from anybody? Otherwise, we will conclude today's session.
Yes, my apologies, guys. It is my fault that I have taken too much time.
So I think no more questions for today. অনেক ধন্যবাদ অভিজিৎ দাসকে মূল্যবান তথ্য নির্ভর ও মনোগ্রাহী বক্তৃতার জন্য আমি অভিনন্দন জানাচ্ছি। এখন আমরা শেষ পর্বে এসে পৌঁছেছি। আমি বিজয়নারায়ণ মহাবিদ্যালয় প্রাণীবিদ্যা বিভাগের সহকারী অধ্যাপক ও বিভাগীয় প্রধান ডক্টর সরোজ কুমার ঘোষ মহাশয়কে ধন্যবাদ জ্ঞাপন এবং অনুষ্ঠানটি আনুষ্ঠানিক সমাপ্তি ঘোষণার জন্য আমন্ত্রণ জানাচ্ছি।
Thank you, Dr. Ghosh and Dr. Manavij Mukhopadhyay. I am very grateful to you, to Sam also, for inviting me and giving this opportunity to talk in front of these students. I'm always feel very good to come back to Burdwan, and this is one of the opportunities that arose, and I'm very much thankful to this organization and to the people who have organized. Thank you, all of you, and to all the students, my thanks. And I didn't notice a lot of questions, or no. Okay, so my email address have been given. I think you can contact me if you have further questions. I'll try to solve them by email. And some of you have requested for study material. I can happily share the PowerPoint material over here. Can I send it to someone? Like Sam, can you coordinate by? I can send the
Yes,
PowerPoint
please.
material that I have.
You may send it to me so that I can post it to their respective group.
Okay, fantastic. Okay, thank you. I think I will